Hello, everyone, and welcome to our webinar that's offered jointly by the Mobilize and FAIR Centers at Stanford University. My name is Matt Petrucci. I'm the Scientific Program Manager of both these centers, and I'm excited to serve as your moderator today. Today's speaker is Parker Ruth, who will share his team's process and best practices gained from the past 3 years of a large-scale OpenCap data collection for rare neuromuscular diseases. In the first part, he will describe the latest findings from their large-scale study of neuromuscular disease biomechanics. In the second part, he will share a process and best practices for planning similar large-scale human movement data collections with OpenCap, including study design, logistics, and data management and sharing. Today's webinar is brought to you by the Mobilize and FAIR Centers, which are both supported by the National Institutes of Health. The Mobilize Center is focused on developing and disseminating state-of-the-art biomechanics and machine learning tools for researchers to analyze human movements. The FAIR Center was recently launched to develop and disseminate AI models to improve outcomes for conditions that limit mobility and participation. Now, before we get started, a couple quick reminders about the webinar format. We will have a research talk and a best practice session. We will take your questions at the end of the research talk and the best practice session. Please type your questions into the Q&A panel in Zoom and not the chat. Feel free to ask questions as he is talking. And one other thing to note, this webinar will be recorded and posted to our YouTube channels. Now I'd like to introduce our speaker for today. Parker Ruth is a fifth-year computer science PhD student at Stanford University. As a member of the Neuromuscular Biomechanics Lab, Parker has co-organized 3 large-scale OpenCap data collection events to create the largest biomechanics data set of rare genetic neuromuscular disorders. His graduate thesis is on designing health sensing tools and digital biomarkers for neuromuscular and cardiovascular health. Parker earned bachelor degrees in bioengineering and computer engineering from the University of Washington in Seattle. His PhD is supported by the NSF Graduate Research Fellowship, an NIH F31 Predoctoral Fellowship, and a WUSI Human Performance Alliance Agility Project Seed Grant. With that, we are excited to have you all here today. I know we are all excited to hear Parker speak about his research, so I'll let him go ahead and begin.
Thank you, Matt. Let me share my screen and my slides. All right, well, let me jump right in. So tools like OpenCap that allow us to do markerless motion capture have already started to become game changers for the way that we collect biomechanics data sets because they allow us to scale up and do larger scale data collections. So in this presentation, I'm going to tell you about how we've used OpenCap to try to advance the discovery of new biomarkers for neuromuscular disease. And then we'll also tell you about how we do it behind the scenes and help you conduct your own larger scale data collections. Before I get started, I want to just set the stage with what we're talking about today. So let's talk about measuring human movement. There are, of course, many different ways to measure human movement in clinical practice and in clinical trials. Some of the most common are called timed function tests, which are measurements of how long it takes to perform a simple task that a clinician can measure just using an ordinary smartwatch. So for example, illustrated here, this person's performing a timed up and go task where you stand up from a chair, walk 3 meters, turn around, sit back down again, and the clinician will measure how long it takes to complete the entire task. These are fairly repeatable measures. They're very widely used. Sometimes they're even used as outcome measures for clinical trials. But the clinical colleagues who we work with here at Stanford Neurology have taught us that just the time alone is a very limited measurement of the overall quality of your movement. So for example, they can see the types of compensations that you have or the parts of the activity that are slower or faster for you. And right now they can only encode those using clinical notes. They don't have a way to quantify those kinds of details. So just to drive that point home a little bit more clearly, I want to share some videos. These 2 participants from our own research have agreed to allow us to share their videos to show you what this looks like. They're both going to perform the timed up and go task. I'm going to play the video a couple times. The first time, I just want you to pay attention to how long it takes them to perform the activity. So you can see these 2 people took almost identical amounts of time to perform the task. So they would get pretty much the same stopwatch score. I'll play it one more time. This time, pay close attention to how they move their bodies. So look, for example, at the lengths of their strides, their cadence, how high they lift their feet off the ground. And you'll see a lot of differences in the way that they moved. So trained physical therapists, occupational therapists, neurologists can immediately see those kinds of qualitative differences. And we think this is, in some ways, can be a limitation of existing time function tests, that by reducing the complexity of human movement down to a single number, they're also missing a lot of these important characteristics. So OpenCap, of course, works differently. We can use a smartphone camera to scan in a participant and then record them while they perform the whole task. So now, instead of just capturing a single score, we get the full 3D motion of their body. And now when they perform our timed up and go task, we can actually record all of their marker positions and kinematics and save that data to analyze more completely. So the question is, can a tool like OpenCap help us advance beyond the capabilities of existing time function tests? For those of you who are new to OpenCap, very simply, the way it works is you set up one or more smartphone cameras, record videos of human movement, they get uploaded to a cloud server, and there they get processed and automatically converted into both marker positions, so the XYZ coordinates of those key points on your body, and also kinematics, so the angles of your joints over time as trajectories. As Matt mentioned, this talk has two parts. The first part will just share an update about the research that we've done and explain how we've done large-scale data collections to create a database for neuromuscular disease. We'll break for Q&A, and then after that, we'll talk about best practices for how you could do this yourself. All right, so let's jump in. The research update part I'm going to break into three sections. We'll talk about the clinical need we're trying to address, how we collected the data, and what we've discovered so far. So the clinical need, we're focused on these rare genetically defined neuromuscular diseases. The reason why we picked these, and I've listed some of the ones in our data set here on the slide, is that we're focused on diseases where we think new measurement tools could really make an impact. Because many of these diseases are pretty rare, there aren't a lot of validated clinical measures that assess their movement quality. So they either have to reuse measures that were validated on different diseases or use general purpose measures of human movement that aren't specific to the unique patterns of movement. And each one of these diseases has a different sort of signature of movement because they affect different muscles and in different rates in different ways. Another important point here is that because they're genetically defined diseases, there are a lot of upcoming genetic therapies that are in clinical trials currently or upcoming clinical trials, and there's a real need for having good outcome measures to advance the success of those trials. But one thing that can make this really challenging is that many of the diseases on this slide progress very slowly, and that causes some real challenges for doing clinical assessments. To show you what that looks like, in a typical clinical trial, you track some kind of clinical outcome. Perhaps that could be your walking speed, for example. And over the course of the trial, you'll see a decline in walking speed as the disease takes its course. And the hope is that the people in your therapy group have a slowed decline or even halted or reversed decline. What makes this challenging for a slow progressing disease is that the course of disease progression can take 10 years or more. And so during the duration of a 1 to 2 year clinical trial, you have to have very sensitive outcome measures that can detect the early stages of that progression. So how do we measure human movement in a clinical trial? A very natural place to start is just to ask participants about their own lived experience. And in some ways, this is a great approach because it's very much in line with what we ultimately care about in terms of the impact on human health. So for example, you can give a person a list of daily tasks like, taking a shower or getting into a car and ask them, are these tasks easy, difficult, or impossible to perform? One potential challenge with using this as an outcome measure is that they can be quite subjective. So just to illustrate that point, here's some of our data from our own data set. We asked participants in our data set to fill out this survey. The person on the top here reported that running is easy for them, and this is how they moved their body at their maximum ambulatory speed. And then the person below reported that running is impossible for them, and this is how they were able to move their body. So we don't exactly know why these were different, but one potential explanation is that the person above, perhaps they define running as any gait speed that's faster than their normal comfortable walking speed, and they find this motion easy. Maybe the person below used to be a competitive sprinter and they consider this a jog. Or maybe they interpreted the question as meaning going on a run around their neighborhood, and maybe they're perfectly capable of running for 10 meters, but they aren't able to go for a run. So this kind of subjectivity limits the use of these surveys as sensitive measures that directly measure the impact of a drug on the way you move your body. An alternative is to just grade those same kinds of activities with a clinical expert to try to become more objective about it. So you can create a battery of activities that a person can perform right in front of a trained clinician who can rate them on the same kind of three-level scale. And in fact, this is also pretty commonly done and sometimes used as outcome measures for clinical trials. The problem with this is that it's very quantized in the degree of sensitivity you have for change. And in fact, this impacted a recent clinical trial for Duchenne muscular dystrophy. There was a drug called Elvitus, and they explained in the trial results, because they weren't able to detect a change during the time of the trial, that a score of 1 on this type of clinician-graded score can represent a broad range of abilities. A person might be able to perform a task with slight difficulty, and that would score a 1. Someone else might perform the same task with great difficulty, and they would still score a 1. And so they found in this study that that assessment just may not have been sensitive enough to detect that difference during the trial. And that's why many people use time function tests, because they're continuous measures of function rather than discrete scores. Of course, we've already talked about the key limitation of time function tests is that they have this very reductionist view of measuring human movement. And in fact, the videos that I showed you before were different because the two people in these videos have two different neuromuscular diseases. The person on the left has a disease called facioscapulohumeral muscular dystrophy, or FSHD. The person on the right has myotonic muscular dystrophy, which is abbreviated DM. And I'm going to be talking more about these two in our results soon. The key piece that's missing here is that a person might walk at the same speed, but be compensating more severely as their disease progresses. And so to really sensitively detect whether the disease is progressing, we have to not only capture how fast they move, but the way that they move. And the way that the disease progresses is different for different diseases. So that's the clinical need we're trying to address. Let's talk about now how we've used large-scale data collections to try to address this need. We picked a set of 9 activities, including 4 common time function tests. And in our study, we wanted to compare these using both the stopwatch scores and also our OpenCap assessments. So our study setup looks like this. We have multiple cameras. Our participants perform the tasks and do simultaneous measurements with our OpenCap video-based biomechanics and also clinical assessments with time function tests. So that allows us to simultaneously take those 2 measurements and compare them directly. Here's what that data looks like once we've encoded it in OpenCap. You can see we have both the raw videos and the skeletal kinematics. We've done data collection in 3 different settings. We started by rolling out data collections in regular clinic assessments when patients come to the Stanford Neurology Clinic and see the experts there. We also started doing data collection at conferences, national and international conferences, and even community events like support groups for people with rare diseases. But what has really helped us to scale up our data collection is doing high throughput data collections on the Stanford campus. And when we put all these 3 together, we've been able to collect a really large data set. So far, we have over 450 participants, many of whom have come back for longitudinal measurements, and that's given us well over 5,000 recordings of human movement. And for many of the diseases here, actually, I think for many of these diseases, these represent the largest existing biomechanics data sets for those diseases. So the way that actually works when we do these high throughput data collections, we reserve space. In our case, we've enjoyed reserving space in the Stanford gym, where we set up many different parallel capture stations and then bring in multiple participants at the same time to sign their consent forms and fill out surveys, and then perform our measurements with trained clinicians and OpenCap operators. So this has really helped us to collect data much more quickly and in a very standardized way than we would if we were either doing clinic assessments or trying to bring people into a traditional biomechanics capture lab. At this point, I want to take a quick intermission just to talk about the team who makes this possible behind the scenes. Of course, we have an amazing set of engineers who designed the OpenCap system and who helped to maintain and support the engineering and the data analysis on the engineering side. We also have a terrific team of clinical research experts in the neuromuscular space over in Stanford Neurology who help us to infuse their domain knowledge into the algorithms, design the experiments. And then we also have a terrific team of research coordinators, not all of whom are shown here, who help us to recruit participants and maintain the very large and heterogeneous data sets that we produce doing this kind of research. So it really takes a village to do this kind of work, and we'll talk more later about the kind of personnel roles that are involved. Once we collect the data, we now have to convert this high-dimensional human movement data into some meaningful measures of human movement that could be useful for outcome measures for clinical trials. And one of the ways that we've approached doing feature extraction is by asking the clinicians to describe what they see. So for example, we would play the same kinds of videos we've been looking at today and ask a clinician to describe what the differences are between those types of movement, and we try to translate those clinical observations into scores that we could automatically compute from their kinematics. And that gave us a set of 34 features that we studied for our research. All right, with that, I'm going to transition into telling you about some of our preliminary findings, and then we'll take a quick break for questions. So the first finding, perhaps unsurprisingly, is that OpenCap is able to reproduce what we measure with time function tests. So because we can directly compare the stopwatch scores and the videos, we can plot them to make sure that they agree with each other. So when we try to automatically extract time function tests from our clinician-graded TFTs, we see they agree very closely. That's true for the timed up and go task. In fact, it's true for all 4 of the time function tests that we studied. This is encouraging because it means switching to OpenCap, we're not losing any information by going this direction. And additionally, OpenCap also helps us to measure disease-specific movement patterns that time function tests can't. So remember, we were talking earlier about how FSHD and DM show different signatures of gait. And in fact, we see no differences in the walking speed between these 2 groups of participants, but we do see changes in their stride length and in the ankle height during swing. And one reason for this is that FSHD is known to affect a gait pattern called a foot drop, where weakness in the dorsiflexors cause your foot to drop as you step and makes you have to lift your foot higher off the ground. And so these findings are consistent with what we know from the clinical presentation of the diseases, and it shows that using a tool like OpenCap allows us to measure something that is missing in just the speed alone. Because OpenCap can detect those kinds of unique signatures, it also helps us to distinguish between the diseases. So we did a test where we tried to classify DM versus FSHD just based on the biomechanics features. And we found, looking at the video-based features, that set of 34 heuristic features we extracted, we could classify those 2 diseases with 86% accuracy. Whereas just using the time function scores alone, we achieved an accuracy of 50%, which is just random chance. And so that's showing us we have the ability to distinguish these unique types of movement. Of course, distinguishing those 2 particular diseases itself is not a specific clinical need we were trying to address, but this shows that OpenCap is encoding useful information about disease-specific presentations. Another interesting finding we had was about being able to shift from these sort of ordinal scales to more continuous quantitative measures. So I'm showing you here a task that's an assessment of upper body motion. The clinician asks the participant to raise his arms over his head with his elbows straight. And you can see, because he has some upper body weakness, he has to bend his elbows to raise his hands over his head. On this particular clinical scale, he would score either a 4 or a 5, depending on whether he can raise his hands over his head. And we developed a continuous score that tries to capture the same sort of construct, but without these ordinal scale buckets. So the basic idea is that we measure your shoulder angle and your elbow angle. Ideally, you would want to have a high shoulder flexion or abduction and a very low elbow flexion. And so in the denominator there, you see we have 90 degrees in the denominator. So in the perfect case, you'd have 180 degrees of shoulder flexion and 0 elbow flexion. So the maximum score would be 2 on this scale. And now between FSHD and DM, we see differences. This isn't totally surprising because we know FSHD tends to affect the upper body more severely than DM does. And additionally, we can also compare our video extracted angle to those clinician-graded upper limb scores. And we see there's general agreement. People who scored higher on the clinical scale also scored higher on our continuous score. And we're also able to detect these differences within a score bucket. We can see the variability in function in those groups. So the hope is that a tool like this might help us to create more continuous and ultimately more sensitive measures of upper body function. A final observation here is that because we extracted these features with input from clinicians, they make the predictions more interpretable. So I mentioned FSHD affects the upper body more and DM affects the lower body more. And so when we do feature importance measures to see which features are most important for predicting FSHD, it's not surprising that some of the upper body features are the most important measures for that disease. Whereas for myotonic dystrophy, we see the most important features are all lower body features. So the remaining work we have to do is to take this set of sort of exploratory features and convert it into a single digital biomarker that's tailored for an individual disease. This is still very challenging work, and it could be that instead of just using these heuristic features, there might also be other approaches using more data-driven methods, going straight from kinematic trajectories to richer embeddings. And we're approaching this both from deep learning methods and also from more traditional statistical learning methods right now. But the roadmap moving forward is to take this rich information that we know contains lots of useful information about human movement and create useful clinical outcome measures or biomarkers for them. With that, I'm going to take a pause and we can answer a few questions.
Great. Thanks so much, Parker. We'll go ahead and start the Q&A. So again, if you have questions, please go ahead and type them into the Q&A. If we don't have any questions come in, we can also just move to the next session and then just have a longer Q&A at the end. Looks like none have come in so far. I guess maybe just to expand a little bit on how much back and forth was there in terms of with the clinician team in finding these features that you wanted to focus on? Maybe you can talk a little bit about that process a little more and how that evolved. Yeah.
Yeah, for sure. The clinical team was very involved from the very beginning. They helped us pick the set of 9 activities because we talked with them about how we knew these diseases would present and which activities would help us to isolate the specific movements we'd be looking for. And then at the time that we started our data analysis, we sat down with them and played a lot of the videos that we saw from our data collection and asked them to speak aloud about what they were thinking when they witnessed them. So, and then as we started doing our analyses, we kept them in the loop with our preliminary results and asked for their interpretations as well. So it was very helpful for us because human movement is such a high dimensional data set. When you do dimensionality reduction, it's useful to come in with some domain knowledge instead of expecting all of that to be data driven.
Definitely. Let's see. We do have a few questions that came in. Cyril Thomas was just mentioning the use of EMG could potentially add accuracy. I guess, was there a consideration of doing OpenCap plus additional sensors or?
Yeah, it's a great question. So OpenCap is just one way of measuring human movement. Of course, there's also inertial measurement units or accelerometry. There's EMG and force plates, a host of other things. In our case, we were just focused on kinematics, but we know there are other research studies in our lab and in other labs that have looked at fusing together the information from multiple.
Definitely. Let's see. G Chen had a question. Oh, well, okay. There's a question about synchronizing force plate measurements in EMG. There are ways to do that, and actually encourage you to the OpenCap forum. There's been a few threads on that if you'd like to check those. A question from Peter Aldana. Are the movement-related biomarkers that you've captured, do you think they could potentially help us understand cognition or higher level abilities of the participants?
It's a good question. So I guess the short answer is maybe. Part of the question there is whether or not you want your measures to capture those things. So, it depends on in our case, because we're specifically focused on designing outcome measures for clinical trials, we really want to measure the effect of a therapy. And so if the therapy affects on the musculoskeletal system, then we want to measure that. And so, but for sure, in terms of diagnosing a disease, the full disease diagnosis about more than just the way a person moves. It might also include a cognitive assessment. Another piece that's missing there is even something like the way a person's face looks. Some of the diseases we're studying affect muscles of the face in distinctive ways, and trained clinicians can recognize diseases by the way that it presents in a person's face. That's not something OpenCap measures, but it could be a useful tool for a diagnosis as well. So OpenCap certainly isn't a comprehensive way to characterize a disease, but we think it is very good for measuring how diseases impact the way you move your body. So that's our focus.
A question from Aubrey Grove. Is the detection best tuned for lower body, or is it also well tuned for the upper body? For example, you mentioned shoulder angle is measurable. Is slight shoulder hiking detectable with OpenCap?
Yeah, it's a great detailed question. So OpenCap is useful for many different kinds of motion. We've had success using it for both the upper body and the lower body. Actually, in the second part of our webinar, we'll talk a little bit more about how you choose what measures are appropriate and whether OpenCap is accurate enough for your particular use case.
Definitely. Okay. Juliet Blackheart has a question about using OpenCap in the EU. Again, that's another one I would. Yes, there are GDPR requirements, and I think there's some forum threads about that as well. Um, and then Naomi Frankston can you explain or can you speak to the accuracy of using OpenCap with a single camera? There is OpenCap binocular that can be used for a subset of these tasks, but maybe we can talk about the accuracy there.
Yes, absolutely. So monocular open cap is pretty new. We're excited that that's a new feature now. In general, as a rough rule of thumb, having more cameras is probably more accurate, but it depends on the specifics of your task. And also, you correctly pointed out that it might be more or less accurate for different tasks. So one of the advantages of having multiple cameras is that you reduce the amount of time that a part of your body is hidden behind another part of your body. So you get more information that way. And so, for example, if you were just using monocular video for frontal plane imaging of a task where you're only doing arm abduction, say, where all the motion is in the plane that's visible by the camera, it might be a very suitable tool. But if you're trying to do a more complex movement and you wanted a certain level of precision throughout the whole range of motion, you might want to use more cameras. That's just a rough rule of thumb. At the end of the day, the best way to know is to test it yourself and see what works.